Hey guys, we are talking about the last two sections of Chapter 9, or cellular respiration, where it's kind of the potpourri of the unit. So we're talking about bacterial respiration, anaerobic, aerobic respiration, and fermentation. And so let's start with bacterial respiration. So bacteria can undergo respiration both anaerobic and aerobically. And so this shows that the process of respiration is ancient because it's been happening since before there were mitochondria. And so glycolysis occurs outside of the mitochondria, and that's more evidence that it's been occurring since before mitochondria have been around. And bacterial cells possess many of the same enzymes used in eukaryotic respiration. And so that tells us that those enzymes came from, originally came from this bacterial process and have been passed on as the cells have become more complex over time. And so the electron transport chain is formed in the plasma membrane. And if oxygen is present in the process or in the environment, it will be used to pull down that electron. And that electron, as you know, is going to attach to water and we'll breathe it out, or it'll be removed from the process. And if there is no oxygen present, well, then another electronegative source will be used. An example is a sulfate would be used here instead of oxygen. It's not as electronegative as oxygen, but it's still good enough. And so when the sulfate is reduced to hydrogen sulfide, it has that rotten egg smell. If you've ever been walking around in mud and you get your foot, you pull your foot out of mud and you can smell that rotten egg smell, that's hydrogen sulfide. That's an anaerobic environment where bacteria were existing because it was under the mud. There's no oxygen there. This also occurs in thermal vents and at the bottom of the ocean. So this is anaerobic respiration. Fermentation is another way to produce ATP in the absence of oxygen. It can occur in bacteria, fungi, animals. So we can go through fermentation as well. If you're working out really hard and we use up all of our oxygen, we'll produce lactic acid. But let's get there in a moment. So fermentation occurs without oxygen or the use of the electron transport chain. It uses substrate level phosphorylation. And if you remember what substrate level phosphorylation is that it's simply phosphorylating an ADP into an ATP by being the substrate. It is the substrate in an enzyme, and it just claps them together, and that is how the ATP is produced. And so in this process, through glycolysis and fermentation, we're going to be using substrate level phosphorylation. And it requires a constant flow of NAD plus to be reduced into NADH. And so there are a number of different types of fermentation, but we're going to be looking at primarily two of them. We'll be discussing lactic acid fermentation, which is what we go through, and alcohol fermentation, which is what produces beer and wine. So lactic acid can be used by bacteria, fungi. It makes dairy products, hence the lactic acid. So we use it when we use up all of our oxygen. So if we are working out really, really hard and we don't get enough oxygen from our blood into our cells, we're going to start using lactic acid fermentation. It's not as effective as cell respiration, but it's better than cell death. And so once there's no oxygen, cell respiration will shut down and fermentation will take over. And so here's the process. This is kind of a weird diagram to wrap your head around, but it makes sense once we do this. So here's glucose, and it's going through glycolysis. And so remember that glycolysis exists outside of the mitochondria. And so glucose goes into glycolysis. Do you remember the 10 steps? If you remember that there was the phase where we had to spend 2 and then we made 4 with a net of 2 ATP. So we went through glycolysis, we've made 2 ATP, and at the end of glycolysis we have 2 pyruvate and NADH. So pyruvate is then going to accept the extra hydrogen from NADH because NAD plus is what fuels glycolysis. We're not using the NADH in the electron transport chain, and so it's just sort of there. It's not going to be used, and so we need to oxidize the NADH into NAD plus so that it can fuel glycolysis. And so what is going to do that is that we're going to add those hydrogens to pyruvate to make lactate, which then becomes lactic acid. And so the lactate is the product that we get from trying to get the hydrogens off of NADH so that NAD plus can fuel glycolysis. And so that is lactic acid fermentation. And here, fungi and bacteria use alcohol fermentation, and this is very similar. So here we have glucose going through glycolysis, 2 net ATP with the product of 2 pyruvate. But then here we can see that the carboxyl group is going to be released as carbon dioxide. So carbon dioxide is produced in alcohol fermentation. And so with the CO2 gone, we now have an acetaldehyde. The acetaldehyde is then used as the hydrogen acceptor, or it is being reduced into ethanol. Ethanol is the alcohol. So OL, when something ends in OL, that means it's an alcohol. And so ethanol is the alcohol that we find in beer and wine. And so that is simply the molecule that is being formed because it needs to accept those hydrogens so that NAD plus can fuel glycolysis, which is a pretty cool little process if you think about it. And so catabolism is more versatile than what we've been studying. So through cell respiration, we've only been looking at the breakdown of glucose. However, we eat more things than just glucose. So if we eat carbohydrates like starch and glycogen, these are going to be broken down into glucose, and they're going to enter the process right at the beginning. So sugars go into glycolysis at the beginning. And proteins are going to, when you eat a protein, they're going to be used for fuel. Some parts are going to be used for fuel, but those amino acids are going to be used as parts. And so those parts are going to be stripped down first, and that's called deamination. And so what's left over after deamination is going to go into the process, either at the pyruvate processing or into the citric acid cycle. So, or at the very end of glycolysis, essentially. Because proteins are so diverse, you're going to get a number of different molecules that are going to go in. And so some will go in at the end of glycolysis, some will go in at pyruvate processing, and some will go in at the citric acid cycle. And so remember that proteins have nitrogen. And so the nitrogenous wastes are going to be converted into ammonia and urea and excreted out of the body through the kidneys, which you know all about. And so finally, let's look at fats. As fats are eaten, we're going to break those down into the two parts. We know that triglycerides have that glycerol head and the fatty acid tails. And so the glycerol head is going to go into glycolysis, and the fatty acid tails are going into pyruvate processing. And they're going to become part of acetyl-CoA. So if you look at, this is an acetyl-CoA molecule. And so here's your acetyl group. This is your acetyl group. And the carbon hydrogens are going to be used in a process called beta oxidation. So 2 carbon fragments are going to be used at a time as they enter the citric acid cycle as acetyl-CoA. And so this is why fatty acids have only even numbers of carbons in their fatty acid chains, because they're used 2 at a time. And so for our last slide, we're going to look at how respiration is allosterically regulated. Let's go back to our original cell respiration diagram and think about that. Let's think about original cell respiration. And so how is cell respiration regulated? It is allosterically regulated. So if we remember that allosteric regulation is when a cofactor or a coenzyme, something, is going to bind allosterically or not in the active site of an enzyme. And that's going to either inhibit or it's going to activate. And so phosphofructokinase, this is our third step in glycolysis. This is our enzyme that does the work in the third step of glycolysis. And it will be inhibited if there is lots of ATP outside of the mitochondria. So if there's plenty of ATP, it's saying, let's not waste our energy and make more ATP. It's going to allosterically bond to the phosphofructokinase. And then when there's not enough ATP, you're going to have an abundance of, you would think ADP, because that would make sense, but nothing in biology is so simple. We have AMP, or adenosine monophosphate, which is a building block of ADP. So when you have lots of this part that needs to be added to make ATP, so when you have lots of these broken down ATPs, the AMP will allosterically bind to the phosphofructokinase, saying, hey, there's lots of stuff, there's lots of building blocks out here. Let's start working, respiration. And so that's going to stimulate the process. So it's going to allosterically bind to the enzyme and activate it. All right, and so that is all of this section. I hope you have a great day.